This poster describes strategies and techniques that we have developed to support the preparation of complex health datasets. The preparations of such datasets is a challenging and time-consuming task. We have developed methods to combat this along two approaches: quality control and semantic enrichment. The benefits of these approaches are demonstrated on a multimodal colon cancer dataset, COLO661. Additionally, an R package is in development to share these methods. The quality control methods, shown in the center of the poster, aim to programmatically bolster the reliability as well as the machine interpretability of the dataset. In Figure 1, the workflow methods applied to COLO661 is shown. The ordering of these steps represents dependencies where later steps benefit from earlier steps. In our R package, users can carry out these steps individually or collectively within wrapper functions. There are several clear benefits of these methods in the case study dataset. These are summarized on the left-hand side of the quality control box. Figure 2 shows a completeness heat map of the dataset, where yellow cells represent missing values and blue cells represent present values. This visualizes the structure of the completeness in the dataset and reveals several regular shapes. These regular shapes suggest a non-random cause of the missingness and help further characterize the dataset. There were several free text variables in COLO661, and so we built on the quantdata package to extract structured information from these unstructured variables. These structured variables recorded the presence of a word or groups of words which appeared near each other in the free text. This was able to extract relevant information on medications and family history of cancer. Overall, these methods produced a high quality and fully numeric multimodal dataset which is ready for machine learning applications. At the bottom of the poster, we can see the process of semantic enrichment. This is applied to COLO661 following quality control. Variables in health datasets are unlikely to be fully independent of each other. For example, diseases which affect a common system or organ in the body, or genes which are part of the same pathway. The process of semantic enrichment uses medical ontologies to extract these relationships and create new variables which contain this information. An overview of how this process works is shown in Figure 3. On the left-hand side of Figure 3, we can see the three required objects: the dataset, an ontology, and a mapping file which will link entities in the ontology with dataset variables. It is likely that groups of variables will share multiple common ancestors. Therefore, we calculate the information content of each ontological entity. The most informative common ancestor is then selected to represent the group, as it is the most specific common ancestor for these variables. Each set of variables are then aggregated and added to the dataset. Figure 4 shows an example of how this process was applied to COLO661. The 5 variables in purple are mapped to the entities in SNOMED CT, which are coloured green. The most informative common ancestor of these entities is heart disease. With this information, these variables are aggregated and labelled with heart disease. They're aggregated using 5 functions to capture the various possible stacking of effect size. These aggregations are then appended to the dataset. In the case study dataset, 93% of variables were mapped to either SNOMED CT or the gene ontology. The variables which weren't mapped were negative findings or missing values. SNOMED CT facilitated the creation of 800 aggregations, whereas the gene ontology 985 aggregations. This represents a significant increase in the information present in the dataset. Thanks very much for listening. If you have any questions, please let me know.